Halo Bu Marcia, there's no voice. Bu Marcia.
We can't hear you, Masha. We can't hear you.
Can you hear me now?
Yes, we can now.
Okay, so sorry about that. Okay, so good morning everybody. Yeah, I'll start again. Very exciting day today because we have Dr. Ines with us today to deliver the talk about vaccines, something that all of us right now anticipated, right? And I'm also happy to see so many participants today in this power talk. So before we begin, I have a couple of code of conduct. So first of all, attendees are not allowed to activate the audio during the power talk session. So keep your microphone muted. And then the second thing, I encourage all of you to ask as many questions as you like using the chat. Yeah, and hopefully all those questions can be answered at the end of the sessions, and I will convey these questions to our speakers. And lastly, there will be the evaluation surveys. It will be distributed after the power talk, and I also encourage all participants to participate in that survey as well. Okay, so now I would like to introduce our speaker, Dr. Ines Atmosukarto. And first of all, Dr. Ines, from all of us here in i3L, thank you so much for agreeing to deliver this power talk today. And it is a pleasure to introduce Dr. Ines. Dr. Ines received her Ph.D. in molecular and cellular biology from the University of Adelaide. She also received a Bachelor of Science with first class honors, also from the same university, where she also did double major, right? Biochemistry and genetic from the University of Adelaide. And Dr. Ines is a high achiever. She received numerous awards, both from Indonesia side as well as from the Australian government side. Notably, she is the recipient of Anugerah Iptek in 2012 and the Australian Alumni Award in 2009. She was also the winner of the prestigious L'Oreal UNESCO Fellowship for Young Women in Science in 2004. And apart from that, she also received many research grants, such as the highly competitive one, that is the Grand Challenge Exploration Grant from the Gates Foundation, and NCRIS and RISS grants from the Australian government. Dr. Ines Atmosukarto is currently the Chief Executive Officer of Lipotek Pty Ltd, located in Canberra, Australia, where she held this position since April 2010. And she is also the research manager of the Australian Cancer Research Foundation, Department of Cancer Biology and Therapeutic at John Curtin School of Medical Research. So with that backgrounds and experience as well as an achievements, it is a huge honor for us to have Dr. Ines as the speaker for the i3L Power Talk today. So without further ado, I would like to invite Dr. Ines to deliver the talk entitled Vaccines, an Undervalued, Underappreciated Medical Product. Will COVID-19, this pandemic, change this? So Dr. Ines, the Zoom is yours.
Thanks, Dr. Masha. That's a very, very kind introduction. When you mentioned the L'Oreal Fellowship, it makes me feel very old because I was in 2004. I was then a young woman. I don't think I am anymore. Anyway, I'm looking forward to giving a little bit of an overview on vaccine development for infectious disease, as well as in therapeutic cancer, and then try and bring at the end of the presentation a little bit of overview of what's happening with COVID-19, because I think everybody's going to be quite interested with what's happening with the COVID-19 vaccines. Now, if I'm going over time, please let me know, Marcia, and I can cut it short and we can go straight into the questions, because I do have a few slides that I've prepared, but we can cut it short. As you said, I spend most of my time with Lipotek, which is a startup company, and it's a niche biotech company that operates out of the Australian National University. In case you missed the John Curtin, Dr. Marcia, here's a picture of the John Curtin. It still looks the same. And we are now on the second floor, on the top floor, where the cancer department is. And as a biotech company, we sit at the intersection between academia and industry, in that intellectual property that's developed at the universities are further developed within the company with the aim of developing products. So the sort of research that we do is a little bit different, because it's very focused on developing product. We specialize on the development of liposome-based vaccines, and as well as using liposomes for the delivery of chemotherapy, as well as immunotherapeutics. And our focus is really on trying to understand the interaction between our liposome delivery systems and the immune system. So trying to understand how we can improve our product by understanding the immunology behind it. So the talk is kind of broad. I'm going to start by giving an introduction to the immune system. I'm going to go over vaccine development and have a look at vaccines for infectious disease, as well as vaccines for therapeutic cancer vaccines. I want to actually speak a little bit about the biopharmaceutical industry and the challenges in actually taking a biopharmaceutical like a vaccine from research to actually being a product. And if we have time at the end, I want to talk about vaccine development in the era of COVID-19. So the next 5 slides are a kind of a really quick overview about the immune system. So hopefully everybody on this talk has got a background in biology at least, and hopefully a little bit of background in immunology. So you probably remember from the immunology classes that the immune system can be divided into broadly 2. You've got the innate immune system and the adaptive immune system. Now, the innate immune system is the immune cells that will react in a short time. So very quickly after being infected, your innate immune system is what kicks in, and it consists of epithelial barriers. Obviously, your skin is kind of like the first barrier that a microbe or a pathogen has to breach. But then if you do have a breach, you have things like phagocytes, dendritic cells, natural killer cells, and the complement, which are the first line of defense, right? So it's the most primitive part of the immune system. It's directed towards very specific molecular patterns, and I'd like to broadly refer to them as danger signals. It has a very broad reactive effect, and it doesn't have a boosting effect, right? And it's not antigen specific. It really recognizes specific patterns in pathogens and raises an immune response to that. Now, the adaptive immune system is the antigen-specific system, which we only find in vertebrates, and it's directed towards specific epitopes. It induces memories, and the effectors are really 2 types of cells. You've got the B cells, which produce antibodies, and you've got the T cells, which turn into effector T cells and will kill the target. Now, what really connects the 2 is generally what we call antigen presenting cells or dendritic cells. These are the cells that would have encountered the microbe initially, tried to get rid of it, and maybe not necessarily have successfully remove them, and therefore now it's presenting part of the pathogen to the T cells and to the adaptive immune system to try and educate the immune system. Now, these danger signals are really important because that's what really alerts the innate immune system that you've got an invading pathogen, and these danger signals or these pathogen-associated molecular patterns can consist of different things. They can be lipids, sugars, peptides, nucleic acid, and small molecules. So these are the things that are going to alert the immune system. Now, interestingly, in vaccine development at the moment, there's a lot of interest in understanding how these molecules are able to stimulate the immune system, because we can actually exploit these danger signals as adjuvants in our antigen formulations, right? And these are what provides the activation signal for your antigen presenting cells, which then, as I said, goes on to activate the adaptive immune system. So what happens during infection? Like in here, I'm using the avian influenza infection. As I said, your first response is the innate response, which will try to remove the assault or the invaders through the innate immune cells, and as a result, you'll get a number of cytokines being produced. Now, if this is unresolved and goes on uncontrollably, this is when you can get things like cytokine storms, right? And we do also see that with COVID-19 infection. So that's an effect of your unregulated innate immune response. Now, as I said, the adaptive immune response will also be triggered, and with this adaptive immune response will come production of the antibodies, as well as the activation of CD8 T cells. And that's been also a point of interest with COVID-19, because there's a lot of discussion around whether T cell responses are also important in our response to COVID-19. Now, if we look at antibody responses, and I'd like to actually look at this slide so that everybody on this talk can actually take away the point that it takes time for your antibodies to actually mount, right? So from the time of exposure to a pathogen, it will be days before you start to get the IgM response, which is the first antibody to generally appear, and then followed by an IgG response. Therefore, and this is what I've been saying since the beginning of the COVID pandemic, using a rapid test antibody response at the beginning of the infection will not detect an infection, and that's why it's really not a relevant screening tool for infection early in infection. Now, these responses can be boosted, so upon a secondary exposure to an antigen, you will get IgM being produced again, and then you will get a bigger IgG response. And in some ways, this is what we're trying to mimic when we develop vaccine. We introduce an antigen so that if we ever become or encounter the antigen again, then we will boost or we will mount an antibody response, right? So looking at the primary and the secondary responses, the secondary response is always faster because you no longer have a naive population of B cells. You've got memory B cells that you are able to reactivate. So if it takes 4 to 7 days to mount the primary response, it will only take 1 to 3 days to mount the secondary response. And the antibodies also become better. So you've got high affinity antibodies. And that's also the reason for boosting, right? So when you do vaccination, you often do a prime and a boost. And that's really to increase the response and improve the antibody response. Now, if we look at T cells, so that's the last part of the immunology lecture. If we look at T cells, you generally have two types of T cells. You have the CD4 helper T cells, and you have the CD8 cytotoxic T lymphocytes, or the CTLs. Now, the CTLs are the T cells that will act like they will go after infected cells and they will kill infected cells, right? So they are there to actually get rid of any intracellular pathogens, which will be hiding within your own cells. Now, the CD4 helper T cells are there to really recognize when microbes are actually hiding within the phagocytes, so within these innate cells, and they're also there to provide help through the production of various cytokines. Now, as I said before, with the antibody response, with a T cell response as well, you have memory being produced with these T cells as well. So with your first infection, not only will you produce antibodies, you will also produce generally T cells in response to your infection. And upon a secondary exposure to the pathogen, then you will also recall these T cell responses and therefore respond not only using antibodies, but also with effector T cells. And in a way, this is what designing a vaccine is all about. It's designing a product that's able to induce, ideally, both antibody and T cell responses. All right, so now we're getting, so that's the end of the vaccine or of the immunology lecture. And now we're going into vaccines, right? So, because I think it's important to understand how the immune response works to actually be able to understand how to design good vaccines. Now, vaccines are a biological product. So that's what really differentiates it from other therapeutics, right? Unlike, drugs, which are generally small molecules, vaccines are biological products. That means that they are actually prepared from a living organism, right? And they are designed to provide active immunity to a particular infectious disease initially, although, as I will try to give you a bit of an introduction, we also try to develop vaccines, therapeutic vaccines against cancer. So a vaccine in the old days really consisted of the disease-causing microorganism, which was either weakened or killed, right? But the new generation of vaccines are a lot more elegant, and we now are able to focus the production of vaccines on very specific antigens. And therefore, the old vaccines, would introduce in your body a large number of antigens because you are actually introducing the entirety of the organisms, while the new generation of vaccines is really trying to focus the immune response on very specific antigens. So I like to call these, more designer vaccines or tailor-made vaccines, which are a bit different to the traditionally made vaccines, which have generally been, developed empirically. I think nowadays a lot more design goes into making these new vaccines, and that's really nicely demonstrated by the type of products that are now in development for COVID-19. So it's all really based on our improved knowledge of the immune response and trying to understand how we can stimulate or better stimulate the immune response. And that's why I highlighted those PAMPs, those pathogen-associated molecular patterns, because to design a vaccine, you need to understand how to stimulate the immune response and also what to stimulate the immune response against. So what's the target antigen or epitopes? So, vaccines are really important, right? So because, and in my title, I was trying to convey that they're very important, but they're very often overlooked. Why? Because generally the actual cost of a vaccine is low. So from a business point of view, they're not as attractive products. But if you look at what they're able to achieve by preventing a disease, we can really look at the economic burden caused by a number of these infectious disease, that goes from billions to millions to billions, and I guess we don't even know really clearly what the impact of COVID-19 is going to be, but certainly it's probably one of the most expensive infectious disease ever experienced by humanity. So having a product that's able to prevent that is extremely important. Now, the components of the vaccines, although before I said, oh, you know, you need something that stimulates the immune system, and we call that the adjuvant, and you need, you know, the active component or the antigen, which is really derived from the actual pathogen. But then, obviously, like every other product, you will have other components in the vaccines that are there to keep it stable and prevent it from degrading. So they are quite complex products. Now, why is it important to rationally design these vaccines, or can we now rationally design these subunit vaccines? And I will focus more on the subunit vaccines. And by subunit vaccine, I mean a vaccine that incorporates an antigen in the form of a protein generally, but it could also be DNA or RNA, as well as the antigen. The vaccine will include the adjuvant and the delivery system. So these are the three components that we think, we as vaccine developers think about when we're trying to develop new products. The antigen, obviously, you need to have the right antigen, so you train the immune response against the correct part or the correct pathogen, and trying to understand what in a complex pathogen, what should you target your immune response against is actually a very challenging task, and various technologies have now been developed that allow you to understand the nature of these epitopes and also predict whether certain epitopes are going to be good T-cell epitopes to include in vaccine design. Now, with your adjuvants, and as I said, I think before at the beginning, I was talking about these danger signals. These are the molecules that are used to stimulate the immune response. And at the moment, there's a very small number of adjuvants that have been used on the market. And I think this is really an area of extreme interest, trying to find new adjuvants to improve how we stimulate the immune response. And the delivery is really, how do you actually present this formulation to the immune system? Because the immune system is actually designed to recognize certain things of certain size and of certain shape. So how we formulate all these things can also affect the actual activity of the product. Okay, so there's a lot of challenges in developing these vaccines, and I think this really the table reiterates what I was trying to say on the previous slide, and by addressing each of these three components, and these are areas of research, of active research, and hopefully maybe we'll see more people in Indonesia being actually actively involved in this research. My group really focuses on the delivery system. I don't develop new adjuvants, and I tend to collaborate with other groups that have developed new antigens. So for example, I'm collaborating at the moment with a group that has a malaria antigen, and what we're trying to do is see if we combine the malaria antigens with different delivery systems, then does it work better, right? So you really need all three aspects. You really need to cover all three aspects to be able to develop better products. All right, so how are they made? How are these different vaccines made? And I think this is a really good overview, because it really also gives you an overview of the different type of vaccines that we see now in the press with regards to COVID-19. So if we look at a traditional vaccine, the way they're made is generally you isolate the virus, you develop methods to grow the virus in a petri dish, or by infecting certain types of cells. So you can either use, Pharaoh cells, or you can use HEC293 cells, and that's really the cell then becomes the becomes the machinery to make more virus, right? The virus which you expand using tissue culture methods this way is then inactivated, and the inactivated formulation becomes your vaccine, right? So this is a very crude way of making vaccines, but it's really the way we've made vaccines the past century. Now, this is how the Sinovac vaccine, for example, for COVID-19 is made. Now, this new platform of vaccines, which we've heard talk about, include different ways of making vaccines. Like first, what you could do is you can use a viral vector. So this is a vector that can infect human cells to express the antigen of choice, right? So you can use like AstraZeneca and Oxford, for example, they use a chimpanzee virus to express a SARS-CoV antigen, right? And that virus, that recombinant virus, is then propagated in vitro in very similar ways and then used as a vaccine. Or you can use just a specific part of the like in this case, like say we're talking about COVID-19, you try to express just the spike protein, or just generally, like for COVID, for example, we express the spike protein, and it is that spike protein that then become the antigen, but it does need to be formulated, as I was saying before, with an adjuvant. Now, this approach is the approach used by the vaccine Merah Putih, which is being developed at the Eichmann. It's also the approach that's used by Novavax, which is another company that's got a vaccine in phase 3 trials. The third or the fourth approach is really using nucleic acids, so either RNA or DNA, as the antigen. So you don't go through the process of producing a protein, you don't go through the process of producing a virus, you just go through the process of synthetically producing nucleic acids, and that becomes then the antigen that's used in the vaccine. And this is the method that's being used by Pfizer and Moderna, which are in the process of applying for emergency use authorization in the US. Now, vaccines can be administered traditionally either intramuscularly, subcutaneously, or intradermally, right? And how you administer the vaccine is actually quite important, because it can dictate how well the immune response is mounted. So if a vaccine has been developed for intramuscular injection, it should not be given like intradermally, and vice versa. Now, something that's of great interest at the moment is to use other ways of administering vaccines that will not require a needle, because obviously if a vaccine needs to be injected, it requires a trained personnel to administer the vaccine, right? And you know, injection comes with cost, because you've got the cost of the syringe as well, and it also comes with biohazard, because you have to actually get rid of these syringes. So something that's been of interest is actually using these nano patches, which is essentially having nano-sized needles which you can apply to the skin, and these nano-sized needles will perforate the skin to deliver the antigen. And at the moment, my group is working with the University of New South Wales to see if we can develop or use nano patches to deliver liposome-based vaccine. And other ways of delivering vaccines is intranasally, and essentially you squirt the vaccine up your nose, and because you have a lot of mucosal surface here, you also induce an immune response. So ideally, a vaccine should not need to be stored at very low temperature, because that will also help or with the distribution. So temperature at which a vaccine storage is also quite important. Now, very quickly, if we look at how long does it take to develop a vaccine, we can look at all these different cases, and you can see that the development of vaccines based on when it was first isolated generally takes years, right? So if you look at the rotavirus one, for example, the virus was identified in the 70s, but the vaccine was only available in 2006. So it's generally taken years to develop vaccines, which is why what is happening at the moment with COVID-19 is really interesting to see, because it really shows what can happen when a lot of investment is made. So these are the different phases that, you know, a vaccine has to go through during development. I mean, the longest stage by far is the preclinical stage, where you do all the research to try and find the best formulation, right? So that includes identifying and producing the antigens, developing the formulation, and then doing the first phase 1 trial, which is a safety trial. And this is where universities generally work. Then from there on, you have the phase 2 trial, which is to test the vaccine in a larger number of participants to see if you actually mount an immune response and how well the immune response. And at this stage, you hope to have reached a proof of concept, right? To show that your vaccine works and develops, you can mount immune responses, and it gives some protection. But this is also still done in a small number of participants. It's only when you take a vaccine to a phase 3 trial that you're actually testing it in thousands of patients, and at this stage that you can actually see the true efficacy of a vaccine. And the same process also goes for the development of other pharmaceutical products. So that it's not just for vaccine. Any drug has to go through this process, which, you know, if you can see, normally takes years, right? Because these phase 3 trials normally take 2 to 4 years. So if people are becoming impatient with the conduct of the clinical trials at the moment, I think we should always put this into context, because what we are seeing at the moment is extremely fast when it comes to clinical trials. Now, I've got like about 4 slides on therapeutic cancer vaccines, and the reason I'm focusing on the therapeutic cancer vaccines is because at the moment, except for Gardasil, which is the HPV vaccines, there's no vaccines that can prevent cancer, right? And the way Gardasil, or the HPV vaccine, works, it actually vaccinates you against the virus that leads to cancer. So with cancer, vaccines have generally focused on the therapeutic aspect, which is, if you have cancer already, can you get vaccinated and train the immune response to recognize the cancer and essentially kill your tumor cells, right? And this is really what, you know, and that's what everybody's trying to achieve at the moment. And I guess I will upfront say that at the moment, there is no product that has successfully done this. And the next few slides are going to try and demonstrate why it is so hard and why it is so important to understand what happens during disease. So, like the other vaccines, the therapeutic cancer vaccines have also used the similar approaches, you know, using RNA, DNA, viral vectors, proteins. All of these have been tried. In addition to that, there's also been cell-based vaccines, where cancer cells from the patient themselves are used to immunize the patient and generate an immune response. So there's been, I mean, if you look at the literature, there's a tremendous amount of literature out there around cancer, therapeutic cancer vaccines, but nothing has really worked very well. And the reason for that is that cancer as a disease is a very complex disease, right? It's not just having tumor cells that replicate, and therefore you just have the same cells, you know, dividing uncontrollably, which is kind of like the simplistic way we're taught about cancer. Cancers, I mean, the tumor is actually a very complex mixture of not just the tumor cells, but also of immune cells in the tumor, in that tumor. And when the cancer develops, not only are the tumor cells dividing, it is also affecting other aspects of your immune system. And I would encourage any students on the talk to, if they're interested in cancer and immunotherapy, to really read up on the hallmarks of cancer. There's not time to talk about it today, but there's various characteristics of cancer and of tumors, which I think are really important to understand, especially if you're interested in developing better treatments, right? Because the tumors actually are able to influence your immune system and therefore promote this sustained inflammation, but they also are able to deploy ways of avoiding destruction by manipulating certain aspects of your immune system. And understanding that, knowing that the tumor microenvironment is actually so complex, because it includes not only the tumor cells, but also immune cells, and it includes immune cells that have been manipulated to not work properly, then it kind of makes sense that it's really hard to develop a cancer vaccine, because essentially your immune system is not working the way it's supposed to be working, because the tumor growth has affected that. And in the last 5 years, there's been a lot of work going in this area, understanding the composition of the tumor microenvironment to try and understand how we can develop better treatment. Now, if a therapeutic cancer vaccine would work, how would it work? It would have to work through T cells, right? You would want, ideally, your cancer vaccine to stimulate T cells so that it goes on and kills the cancer cells, right? But as I said, right, you will need the T cells to be able to infiltrate the tumor. So not only to be activated, but they need to be able to get inside the tumor. And the tumor is really smart in some ways, because it's able to develop ways to prevent that. You will also need to identify the cancer antigens, right? Because if you think about a cancer, it's not like a virus. A virus is foreign. So the proteins that are expressed on the virus are foreign. Now, the cancer is actually your own cells. So the cancer antigens are mostly like the antigens that are expressed by the tumor cells are essentially your own antigen. So there's been a lot of work going around trying to identify whether there are specific tumor specific antigens, because those would be the antigens that you want to focus on. And there's generally two types of antigen. The tumor-associated antigens and the tumor-specific antigens. And again, I'm not going to go into details, and there's a lot of literature about that. But the TAAs are generally prevalent in a number of different patients. So, they'd be common to breast cancer and breast cancer in different people, for example, while some of these tumor-specific antigens are very specific to a patient, right? So this is where personalized treatments might come in, but we are kind of still a long way from that. So harnessing the immune system to fight cancer using therapeutic vaccines, if there was a role for a therapeutic vaccine, would really need to think about trying to address or shift this balance. So the way we see the growth of a tumor, or the division of these tumor cells, it's really a balance. It's a balance between the growth of your tumor and the effect of the immune system. And as I said before, in your tumors, you will have cells that have actually grown to recognize these as being tumor cells and foreign, and they're trying to get rid of it. But there's also cells in there that are actually being promoted by the tumor to prevent your immune system from working. So some of these are these tumor-associated macrophages and these myeloid-derived suppressive cells. Now, if we were to use a therapeutic cancer vaccine just directly, but if these elements are prevalent, then your therapeutic cancer vaccine is not going to be able to shift the balance. So really, for a vaccine to work in the context of cancer, you need to try and reduce these factors and increase those factors. And you can do that in different ways. There's at the moment, there's immunotherapies that have come out there, which have been shown to work in some patients, these checkpoint inhibitors. Oncolytic viruses have been used to try and reduce the immune suppression in your tumors, and that will allow your vaccine to work. So essentially, you want to try and reduce the immunosuppression inside the tumor so that you can induce anti-tumor immune responses. And really, personally, I don't think that is going to be a single silver bullet that fights cancer. No single therapy is going to be able to actually get rid of a cancer totally. What you need and what people have started focusing their attention on is this combination of therapies. You want something that modulates the inflammation. So what makes the tumor immunosuppressive? You want to try and reduce that. You want something that can boost your T-cell immunity, and this is where the vaccines would come into play. And you want to also completely reverse the immune tolerance, and these TGF beta inhibitors and these checkpoint immune inhibitors are shown to be quite important, right? So what I was trying to do, really talking about these cancer vaccines, it's really not as easy as infectious diseases, because the infectious disease is clearly foreign. The tumor has learned to grow within your body, and therefore it's learned to manipulate the immune system. So now that we understand that, we know a little bit about vaccines and how complicated the way they work, the mode of action is, I want to quickly go through a few slides, some of which I've adapted from my good colleague, Dr. Neni Nouraini from PT Bio Farma. And I want to just go through the process of making these vaccines, just so that we can understand, I'd like to convey how complex it is, and therefore, again, trying to put in context what is happening around COVID-19 at the moment. So these biopharmaceuticals really encompass any biological product, and that includes vaccines, and that's because they're made using biological systems. So that could be bacteria, there could be yeast, there could be insect cells, or it could be eukaryotic cells. So these are the four systems that are normally used. Now, obviously, the cheapest way of making something is in bacteria, but not everything can be made using bacterial cells. And the manufacturing process is actually quite complex, right? So first you've got to understand what it is that you're trying to produce, right? If it's a protein, say like an antigen for a vaccine, you've got to clone it into a suitable expression vector. Then you have to cultivate it, so that is to make liters or hundreds of liters of this. And so usually what you'd have to do is then select, this is where you select the system that you're going to use to produce your biological, right? So this is where you're either going to be using bacterial expression or eukaryotic expression. Now, it is the bacteria or the eukaryotic cells that are going to make your protein in this case, and the yield that you get will depend on the nature of the protein, on how big it is, on what type of protein it is, and what system you're using. But then, you'll have to purify it. So this is essentially what is happening at the Eichmann Institute at the moment. They're trying to go through this process, right? And you've got to try and purify it, because your biological product has to be of very high purity. You cannot inject into someone a product that is not pure. And then you have to formulate it, because it's not just good enough to have the pure protein. You need for the protein to be stable, and this is where formulation is important. So you can see how complex all these steps are, and how complex the road to getting a product is, right? Because what I have described in the previous slide is essentially just here, right? It's the exploratory making the material. Then you have to test it, right? Once you've proven in animals that it works, then you have to test it in humans. So again, it is a very complex process that generally requires years. And at the moment, we're trying to do this within less than a year of a novel virus being isolated. And I will just quickly highlight that producing these biological products requires specific facilities, and this is what you call good manufacturing practice type facilities that are able to make things under good manufacturing practice. So you can't just make it into any lab. You have to have very specific facilities. Now, this is a really interesting graph which I've taken from the New England Journal of Medicine, which really again highlights why is it, a lot of people are saying, well, how come we're developing these vaccines so fast? Are they going to be safe, since normally something that can take up to 12 years is only going to be done in 12 to 18 months? And what we need to understand is that the reason that we can actually do this so fast is because the companies that have developed these products have been specializing in doing this. So they have all these platforms ready to go, and they can then input things into the platforms to be able to move the process really fast, so that things take much shorter time to develop. It takes a lot shorter time to do the preclinical work, and the clinical development, instead of being done sequentially, phase 1, phase 2, phase 3, it's kind of done overlappingly. So a phase 1 is a phase 1/2 study, and a phase 2 is a phase 2/3 study. So you can get a lot of information much faster, and therefore compress all of these steps into shorter steps. It's not designed to bypass anything. It's designed to do it at a faster pace, and it's only possible to do it because there's been a humongous amount of investment done in this area. All right, so again, if you look at SARS, which is the cousin of SARS-CoV-2, it took 20 months from when the SARS virus first appeared to starting a phase 1 study. If you look at COVID-19, it took less than 5 months to get to that step. Why was the world able to go so much faster? Because of all the learning that happened during SARS. There was so much information that was available from all the work that was done during in 2003 during the SARS, the first SARS pandemic, that could be applied, and therefore the actual research stage was able to be shortened to develop vaccines for COVID-19. So I think this is now probably, it holds the record for rapidness. So I think I'm now towards the end of the talk. We now have, I think, at least 11 vaccines in phase 3. So I think in August, when I gave a talk earlier, there were 8 in phase 3. There's now 11 that have either completed or have presented interim results or are now in phase 3, and they cover the 4 different types 4 different types of platforms, right? From the inactivated traditional vaccines, and that's generally companies in China and India. And then you have the mRNA vaccines, which have been the first ones to give quite very good results in their phase 3 trials. These are interim results, and we can discuss that later if there's any questions around that. And then you've got AstraZeneca, who is using a viral-based vaccine, who's also recently reported some interim results for their phase 3. And Novavax is at the moment the only protein-based vaccine that's in phase 3 trials. And it's always going to take a bit longer to develop protein-based vaccines because the process is more complex. Now, looking at, and this is now quite interesting because there's so much information around these candidate vaccines for COVID-19, and trying to understand what what's in the media and what's in all the papers. I think it's really important to try and have a look at how each of these trials are being conducted, and how what sort of information they are trying to extract from the trials, and how is the interim analysis going to be done. So just to kind of look at the 3 that have now made announcements, you can see that all 3 companies, and if you go back to their clinical trial protocols, and now I'd like to say that it's quite extraordinary to have so much openness from these companies as to how they're conducting their trials. But if you can actually look at how each of these companies are conducting their trials and how they've designed their trials to try and understand whether or not you can compare what's being done between all these different companies. And interestingly, all of these companies have actually said they will actually give an interim analysis or conduct an interim analysis when a certain number of positive cases, in this case events, are reported in the trial. So, they vaccinated everyone, and at the point where they'll have either 53, 100 or 106 or 151 positive cases in the trial, they'll unblind it and have a look, out of these 151 people, how many were in the placebo group, how many were in the treated group, and that's how they can actually infer the efficacy of the vaccine. Now, interestingly, and I think I'd like everybody to also note that these trials are designed to detect prevention of disease. Most of these trials will actually test for prevention of disease, not prevention of infection. And we can discuss that later. But because they rely on the presentation of symptoms, right, as an event will be a patient that reports symptoms, right, not based on conducting regular swab tests of the 30,000 participants in the trial. So that's why you can't for most of these trials you will not able to determine whether the vaccines will prevent transmission. You will be able to determine whether the vaccine prevents severe disease or disease. Now and I think again I'd like to and I talk a lot about money because money is important in doing research, right? And we can see how much money has been spent on developing this vaccine and how these various countries or organizations like the Gates Foundation has done a huge amount of investment, $2 billion to try and help move the development of this vaccine. So unless you have this sort of investment, it is very difficult to move a product so fast. And I think we all have to take this in mind or keep this in mind when we read what is happening with the vaccine merah putih in Indonesia, right? We have to compare this sort of investments that have been done overseas with the sort of investment that is being put towards that vaccine. And therefore, you know, can we expect the same amount of speed or not? Now, once it's not enough to develop a vaccine, you have to be able to manufacture it. And interestingly, the manufacturing capacity for these vaccines in the world is actually quite limited to a small number of countries, right? And here I'm using data for the influenza vaccine, but generally the companies that are able to make influenza vaccines should be able to make at least some of these COVID vaccines. And you can see that manufacturing capacity is not distributed equally around the world. Certain countries will be the manufacturing hubs for this product, and therefore ensuring, you know, we have enough manufacturing capacity and also ensuring that these products are distributed equitably will be a big challenge for COVID-19. Now, I'll note that Indonesia is part of these countries that are able to manufacture vaccines, and in that way we are lucky because we have Pty Biopharma who specializes in this product. But there's, you know, aside from the development challenge and aside from the manufacturing challenge, there's the storage temperatures, or the logistic challenges as well, because I'm sure that some of you would have read in the papers that some of these vaccines, especially the RNA-based one, require quite low temperature for storage, right? And obviously trying to ensure that the vaccine stays at -70 in a country with limited resources is going to be challenging. So some vaccines are not going to be appropriate for deployment, even in Indonesia, for example. And then even if you are able to make it, produce it, store it, and distribute it, we need to actually have people willing to take it up. And I think this is also a significant challenge, because with all of the information that's available online and through newspapers, I think we have so much information that it can be a little bit dangerous as well. And we've seen the spread of false information occurring over the last 10 months. And I hope that everyone who's on this talk can play their part in actually spreading correct information. So if all of us are able to, you know, educate the people in our vicinity, in our family, in our groups, then hopefully we can ensure that the uptake of these vaccines, when they become available, is not going to be a problem, because we know that we need a significant number of people to be vaccinated for a vaccine to have an effect. And at the beginning of the pandemic, I think a lot of experts have said that we need at least 70% coverage to ensure that we can we can reduce or get rid of the pandemic. So I think this is probably the last slide. How much do we value preventing a potentially deadly disease? And I think that's an interesting question, because I've really put two different types of information on this slide. On this corner here is the cost, and this is, you know, based on the US, the cost of a vaccine, and at the bottom here is a cost of some of these immunotherapies, right? So these vaccines, you know, are able, you know, to prevent what can be potentially deadly diseases. And you can see that even the most expensive vaccine is about $150 a dose, right? As opposed to an immunotherapy, which is made often using very similar processes. So the manufacturing process that I've explained for the vaccine also applies for making these products, and all of these are antibody-based products. And you can see how, you know, if you are an investor, and I'm now talking from the point of view of a biotech company, if you are an investor and Ines goes and tries to get them to give me money to develop a vaccine, and someone else comes to them with the idea of developing an immunotherapy, sadly, very often there will be a lot more investment going towards these cancer-targeted products rather than vaccines, simply because the return on investment is so high. Despite the fact that although these products have been amazing and you know, they've provided care or therapy for some people who before this had no other option, in a lot of cases these you know, therapies only work in 30% of the patients, of cancer patients, right? And you know, as opposed to a product that is able to prevent the death of many people. So I really hope that the COVID-19 pandemic has put a spotlight on vaccine, has put a spotlight on how important it is to develop vaccine products, and also to develop the capacity to quickly respond to a threat and develop these products. And I hope to see Indonesia being much more active in this space and having the capacity to develop this product competitively and rapidly. So I'll just summarize by saying that, you know, vaccines are valuable public health intervention. I think that's no one's gonna argue, unarguable. Vaccine development is, I think, entering a new era, because we're seeing products being developed in very different ways. We're moving away from this empiric development approach to this more tailored formulation approaches. There is a need to be able to rapidly develop products, and it's been highlighted by the pandemic. And I think the world has seen some amazing work being done in less than 10 months. And I think what it shows is really that investment speeds development. If, you know, if money is not an issue, then I think there's enough smart people around to be able to develop these new products. And with that, I think I've reached the end of my presentation. So if there's any questions, I look forward to answering them.
Well, thank you very much, Dr. Ines. That was a really comprehensive talk about vaccine, from basic immunology to vaccine development, manufacturing, and a lot of issues surrounding successful or not successful vaccines development. And from the Q&A, we have several questions here. So maybe I just go from the first one. So about the type of vaccines, which one that will be the most effective for COVID-19?
That's interesting. That's a question that I get quite often. Mm-hmm. I think most of the candidates that are in phase 3 will probably be very effective, right? And at the moment, I think everybody's getting fixated on numbers, right? Because we've seen Pfizer said that they're 90% effective, and then Moderna said that they're 95%, and then someone else says that the reality is that it's not just how effective the vaccine is that's important. It's also how easy is it to manufacture, right? Because if a vaccine is very effective, but you can only make 10 million doses a year, that's not going to be helpful, right? So how easy is it to manufacture? How easy is it to distribute? And how affordable it is? So it's really, it's not like, you know, getting the best mark for your essay, right? It's really balancing all these things, and what is good for a country might not be ideal for another. Because say, even if Pfizer is, you know, gives you 100% efficacy, right? But if it can't be deployed in Africa, it doesn't matter for those people there, right? So really, it's important to kind of look at this vaccine. It's really not a race. And I think I want people to understand that. It's really not rankingan di sekolah, right? It's not like that. It's really, so long as they provide, and the WHO has already given us guideline. The WHO said that anything that provides at least 50% efficacy is going to be suitable, right? So I think we don't need to get upset, say, if our government decides to go with a vaccine that provides 80% efficacy instead of 90% efficacy. It's not putting our lives at risk. It's because so long as all of these vaccines are safe. That's what I'm trying to say. Hope that answers. But having said that, as a scientist, looking at the phase 2 data, I've looked at the antibody responses by all of the vaccines that are in phase 3 trials at the moment, and the Novavax vaccine gives you the highest titer of antibodies. So if you were to look at the data, then the one that gives you the highest level of antibodies so far, based on the phase 2 data, is the protein-based vaccine. So I guess you can look at it that way as well.
Right. Thank you very much. Yeah, we got that question a lot as well, right? The next one is about vaccine from Moderna, because you mentioned there are so many different vaccine formulation, and this one is mRNA. Is this the first mRNA vaccine ever developed?
So both Moderna and Pfizer. So Pfizer is collaborating with BioNTech, which is a German-based startup. They're the two mRNA vaccines. But yes, these are the first. So those two companies, those two mRNA vaccines for COVID-19 are the first ever mRNA vaccines ever being made for or gone through phase 3 and almost ready to be to receive EUA. So yes, there is no other mRNA vaccine on the market before this.
That's interesting, because following this question, like in the lab, when we want to get mRNA, we get like really tiny amount, right? So I was wondering how they will scale this up into kilos.
So all of these mRNA vaccines are synthetically made. So it's chemistry in some ways. It's more chemistry than biology, this one.
Okay, thank you.
I think, yeah. I mean, obviously there's a lot of know-how that's not available to the public, but yeah, all of these are synthetic.
Right. Okay, so the next one is about vaccine but for cancer. So I just read the question. Would you mind explain again about the vaccine that affect the balance between tumor growth and increasing immune system? How vaccine work to inhibit tumor growth where actually they are foreign particle that will be eliminated by immune cells?
That's a good question. So I get it's really complex, and I was trying to really convey some very complex ideas there. So in the tumors, you have the tumor cells and you have your immune cells as well, right? So in the tumor is not just tumor cells. It's a lot of things, including immune cells. And the reason for that is because as your tumor grows and it starts to express some of those tumor-associated antigen and tumor-specific antigen that I mentioned earlier, your immune system actually sees it as being foreign, and it will start to eliminate it, right? So at the beginning, you actually have a balance between your immune system and the tumor. And for a little while, your immune system is probably able to keep the tumor in check. But at some point, the balance tips and the tumor is able to avoid the immune system. So there's in tumor immunology, there's something that we call the 3 E's, right? So you've got the establishment, you've got the elimination, and you've got the escape, right? So these 3 stages which involve the immune system. So when you're trying to develop a vaccine for cancer, a therapeutic vaccine, what you're trying to do is actually stimulate the T cells that are in the tumor so that they can actually go and recognize the tumor cells and kill them. So yes, the vaccine is foreign, and yes, it's going to be eliminated. But in the process of being eliminated, it will stimulate your immune response. And then your immune response will recognize these things, which it sees also on the tumor. And it will then go on and kill the tumor cells, hopefully. The challenge is really the fact that the tumor microenvironment, which I described before, tends to be immunosuppressive. You have a lot of immune suppressive cells in there, which actually stop the T cells from working. So even though a tumor vaccine might educate the T cells to recognize them, there's things in the tumor microenvironment that still stops them from killing the tumor cell. Therefore, it's important to have these combination approaches where you can lower these immune suppressive things so that your T cells can go and do the work. So I hope that answers it.
Yes, it's tricky, isn't it? The tumor cells. But there is still one question here, but still related to cancer vaccine. So the question is regarding the cancer vaccine. Is it more effective than using CAR T cells and immunotherapy? But maybe you can.
Okay, yeah. So someone's done their homework on immunotherapies. So CAR T cells are essentially T cells that are trained to recognize a specific antigen, and this training, I believe, occurs ex vivo. So you have to take the blood from the patient, and it all occurs outside of the body, and then you put this back into the patient, right? So it can be technically quite challenging, right? And the T cells will recognize one specific epitope, right? While a vaccine can be designed to recognize multiple epitopes, and they also, one vaccine is hopefully gonna be, you would want the vaccine to work on many different people, right? So it's not a personalized approach. So will it be more effective than CAR T cells? I mean, if you choose the right CAR T cell for the right tumors, the CAR T cells will be very effective, right? But again, it's very expensive, right? A CAR T cell therapy is hundreds of thousands of dollars, I believe, still, right? As opposed to a vaccine that you're trying to make for a much lower cost. So the CAR T cells essentially prove that the immune system can work and recognize the tumor. And it's the same with these immunotherapies. These immunotherapies at the moment tend to be antibodies that recognize specific molecules on immune cells which inhibit immune responses. As I said, during tumor development, you often have activation of these or expression of these inhibitors, and the antibodies inhibit that, and they're able to then let the immune response work. And again, for the right patient, immunotherapy works very well, right? And we've seen this with melanoma, which before immunotherapies, melanoma patients often had not many choices. And for some melanoma patients, these immunotherapies work very well. However, at the moment, only about 35% of patients benefit from it. So it's still not ideal, right? So, and in some ways, everybody wants to find ways of identifying which patient will benefit from what therapy, so we can give the right therapy, the most optimal therapy for the right patient.
Thank you. So we leave the cancer cell behind. Now we get back to the infectious disease. So you mentioned about vaccine, the COVID-19 is that prevent disease, but not preventing infections. Why is that?
Okay, so in the context of COVID-19, for example, you have a big spectrum of disease, right? You have some people that have no symptoms, some people that have some symptoms like fever, aches, and then you have some people that have very severe disease and end up in ICU and needing, ventilation. Now, at the moment, and what the WHO requirements were, is a vaccine that prevents disease, in that what we want to see is a reduction in the number of people that develop severe symptoms. So that way, the vaccine might not stop you from being infected, but it will stop you from developing severe symptoms. So that's the difference between preventing infection and preventing disease. You can still be infected but not get the disease, right? And the only way that you can tell whether a vaccine prevents infection will be to regularly swab everybody in the trial throughout a specific period to determine whether they're infected or not. And that is logistically quite difficult. So at the moment, the efficacy of the vaccines is really based on the symptoms. So what they're doing is looking in the participants, how many people develop symptoms, how many of those are severe, how many, and out of those, how many were vaccinated, how many were in the placebo group.
Right. So is it that more than how the clinical trial is conducted rather than the
Absolutely. Absolutely. So it could very well be that these vaccines prevent infection. We just don't know it because we don't have the data, right? So this is why it's quite important to understand how the clinical trials are designed, right? Because, and then if someone claims something, you can actually judge whether the claim that they make in newspapers, or for example, is right or not, right? If the trial is not designed to look for infection, then you can't claim that it prevents infection.
Right. And we cannot like extrapolate from the phase 2 study as well, right? If it is neutralizing antibodies,
no,
we cannot.
You can't extrapolate that.
Right. The next one. So thank you very much for the presentation, Dr. Ines. It was very interesting update on vaccine regarding COVID-19. And the question is serotype. Is there any different serotype from not COVID-19 but SARS-CoV-2?
Yeah, so I assume that like by that they mean whether there's different like genotypes, like of if the virus has different types of mutations. Look, that is really not my area of expertise, so I don't want to say anything that's wrong. But what I can say is that based on the information that's out there, there is no indication that any of the mutations that are out there will prevent any of the vaccines that have been developed from working. So at the moment, no one is too worried about that. But it's not, it's not impossible to happen. But there's no evidence that it's something that we should seriously be worried about.
All right. So maybe we move to the next one. So this one related to the OPV, oral polio vaccines, that can cause the virus-derived vaccine poliovirus. But are you aware if there is like a live attenuated SARS-CoV-2 vaccine developed?
I have the Sinovac one, for example, I'm pretty sure is inactivated. And I think Sinopharm as well is inactivated. I'm not aware that there could be. Look, I mean, there's 150 candidates in the pipeline, right? So I've only really been looking at the ones that have made it to phase 3, and I don't think there's any attenuated ones. Because, I mean, there's a lot of issues with attenuated vaccines, so I think manufacturers try to stay away from that.
Right. So the next one we have here is about the things about the adverse reaction toward vaccines. What is your comment about that? Adverse reaction toward the new RNA-based vaccines in the long-term run.
Yeah, so I mean, and you know, because someone before asked whether it was the first product for like whether these are the first mRNA products, and they are. So we don't really have a lot of knowledge about the long-term effect. But we have to also realize that almost any vaccine will have some adverse effect, right? I mean, if you get the flu shot, you will get a sore arm. You know, you might get, I mean, when I get the flu shot, it's always like hot to touch, and like for a couple of days it's a bit hard to move. Those are normal, right? Because you are actually challenging your immune system, right? So a vaccine is really a challenge to your immune system. If you have no reaction to a vaccine, you have to ask yourself whether your immune system is working the way, you know, whether you're mounting an immune response. So, you know, we shouldn't be too worried if we have, you know, some soreness or even a little bit of a fever. Of course, you know, that, you know, so long as it's not serious. But none of the vaccines in phase 3 have had, you know, some serious adverse events. And based on the phase 2 trials, for the phase 2 trials of the mRNA vaccine in doses higher than the ones that are used now, some patients had some significant reaction, and that's why they used the middle dose, right? So that's what the phase 2 are for as well. You in the phase 2 you do dose escalation to try and find what's the dose that you can use. So obviously we don't know of a potential long-term adverse events, but that's why pharmacovigilance, and in one of the slides I probably didn't talk about it properly, but even after a product has received approval to go on the market, they still had to be monitored, right? The companies will still monitor for adverse events, and in some cases, even the regulatory authorities will monitor and keep track of that, right? So there will continue to be monitoring of these adverse events. But because it is a pandemic, right, obviously you have to balance things. With a normal vaccine development, your trials would be 2 to 3 years, so you can monitor these people even 3 years after they've been vaccinated, and you can see, well, you know, even 3 years later, we didn't see anything. We don't have the luxury of the time at the moment because it is a pandemic, right? So it's all about a balance.
Right. Which also lead me to the questions, like, because in the trial we have the placebo group and we have the treatment group. So at some point the placebo group will also be administered vaccines, right? So how do we then compare like long-term effect of a vaccine?
Yeah. So it's and let's say this is an ethical dilemma, right? So which was interestingly last month there was a big panel discussion by the FDA, and they were discussing that because once a phase 3 trial is shown efficacy, the companies are morally obliged to offer to the participants that were in the placebo group to offer them to get treated as a priority, right? So if you've been in a trial and if you had a placebo, you, because of your contribution, right, you contributed to science and to humanity, you will be one of the first ones to be vaccinated. But as soon as that happens, the trial loses power, right, loses statistical power. But it's a more moral obligation, right? So obviously, again, that's going to be, you will not be able to compare to a control group anymore. But they will still monitor, you will still be able to monitor the effects, right? Because patients will still be able, the participants will be able to report any any side effects.
Right. So I think we have 5 minutes past 12, which is overtime. It's very interesting talk, Dr. Ines, very interesting discussion as well. And I think even though I want to discuss it further, also the other student, I think we want to dig more further into this vaccine, and also the technology that you develop in Lipotek. It's also quite interesting and very interesting indeed. But sadly, we have to end these sessions. And on behalf of all of us here in i3L, we would like to thank you for the time spent and for the very interesting talk that you present to us.
Thank you very much for the opportunity, and hopefully maybe there'll be a chance to have like smaller discussions or more targeted discussions in the future. Yeah, I'd like to get to know i3L better in the future.
It is
Thank
mutual.
you very much.
Yes, thank you very much, Dr. Ines. Thank you.
Bye-bye.
Bye-bye. So thank you, everyone. And don't forget to submit your response for the survey. Okay. And for the virology students, as well as the cancer immunology students, please log in or check in into the link that I have sent to you. Okay. Thank you, everyone. Have a nice day. Bye-bye.
Bye.
Bye.